Hi, my name is James Haldane. I'm the team lead for the iPSC platform team within bit.bio. And today I'm going to be talking about the utility of OGM as a technology within the cell engineering pipeline, how we've employed the technology within our company, and how that usage and scope has evolved over time. So to start with a bit of background on bit.bio, we're a synthetic biology company that provides human cells for research and drug discovery. And to this end, we employ the company's proprietary cell reprogramming technology, Optiox. And this involves a safe harbor gene-targeted approach to inducibly express transcription factor combinations that reprogram iPSCs to a range of mature human cell types. And it's through this technology in our platform that we have the ability to create any human cell type and manufacture at scale, both efficiently and consistently. And it's also with this approach that we're looking to empower biomedical innovation and essentially coding cells for novel cures. So bit.bio has an expanding portfolio with 50 products currently on the market and more in the works. We provide wild-type cells as well as derivatives like disease model cells, engineered to contain disease-specific mutations, CRISPR-ready IO cells, which express Cas9 nuclease for rapid gene knockout generation, and CRISPR screens, as well as IO tracker cells expressing reporter proteins. So as I mentioned, the Optiox technology allows us the potential to build any human cell type. And so we're expanding our catalog of available cells more and more over time in regards to the types of cells we can offer for all the product types I highlighted. And inherently with such work, we utilize robust characterization and QC panels for our cells throughout the process to ensure the highest quality and consistency. And it was in exploring the best technologies for this characterization that we initially gained an interest in optical genome mapping. Within bit.bio, I'm part of the iPSC platform team within our manufacturing department. And the goal of our work is to provide fully characterized banks of iPSCs to support the company's range of products and ongoing research needs. And in practice, this often involves culturing established iPSC lines through one or multiple gene editing events and for up to dozens of passages at once. And as part of the aforementioned characterization efforts, my team also runs a dedicated OGM service, which is utilized across the company, which I'll be focusing on for this talk. So due to the nature of the work that we do, it's prudent that we're assessing the cytogenetics of our cell lines on a regular basis. And so it was in 2022 that we first implemented the Bionano Sapphire platform. Up until that point, we'd been using a more classic approach of chromosomal microarray and karyotyping. And we were interested in OGM for the greater reporting depth and potential that we thought it might offer us. And the primary utility of the technology from the get-go for us was as a tool for routine QC of our cultures and to verify and inform clone selection after editing events. And just for context of other users of the technology, these are the analysis pipelines that we currently employ. So de novo pipeline we use after subcloning in order to get the best possible resolution for detecting any novel abnormalities. But we also utilize the low allele frequency guided assembly pipeline, where the higher VAF sensitivity gives us the best capacity to detect novel structural variants that are emerging in subpopulations within our cultures. And finally, we use the dual analysis pipeline to compare lines against basically the previous result of the same line in the pipeline to identify any discrepancies that might have arisen during the time period between them. So now I'm going to talk about the different ways in which we utilize the technology, both those that we initially intended, but also those that we discovered over time as we expanded our usage of OGM. But starting out with the most fundamental aspect, which is the routine cytogenetic assessment. And in regards to routine in vitro culture, one of the main concerns we have are recurrent abnormalities. So novel abnormalities and mutations occurring in the cells that will confer a growth advantage, which will over time persist and expand to make up a larger proportion of the culture. One prominent example is a gain in the 20q11 region, but there are other regions of concerns we tend to look for, some of which I've highlighted in the figure on the right. But outside of this, we're also screening for more substantial abnormalities that can arise in low levels and then be isolated during subcloning events, with one example we had here highlighted where an edited clone had an aneuploidy of the X chromosome. And in regards to detecting such structural variants, we compared our results to the G banding and AGH that we had before, and we regenerated the same results in OGM and found that OGM identified all of the abnormalities that the aforementioned techniques did, and in some cases, other variants that we hadn't otherwise seen. And this routine cytogenetic assessment is what we initially envisioned for our purposes to be an in-house alternative to karyotyping our material. And as the company and the team has grown, so too has our utilization of OGM as a technology to serve other purposes within the company. So following on those lines, one of the purposes is getting insight into our gene editing events that we employ to integrate our Upchuck's technology and generate reporter lines, amongst other uses. And part of this has involved looking at Bionano's 6 base pair label motif within our own genomic reprogramming cassettes. And in this image, you can see an example of an editing event that caused 2 new consensus sequences. And with this editing event, we had the CTT AAG motif at one end of the intended integration sequence. And though this is not a substitute for the resolution we would achieve with genotyping, with the label motif at the start of each cassette and the relative spacing between the new labels, we can infer and validate copy number of these concatenamers quite readily. And in the case of this sample as well, OGM proved very useful as a screening tool for an unintended polyclonality following subcloning, which we would not have otherwise detected by routine PCR. And in this case, we had 2 subpopulations of approximately equal proportion, with the wild type consensus sequence not shown. And of course, the optimal use case of this intentional labeling is to have multiple motifs within your integration to create unique identifying barcodes. And the greater extent to which these motifs can be incorporated into integrations, the more confidence you can have essentially in when they're observed with OGM. And you also have the potential to ideally and also identify partial or off-target integrations that might be occurring. And it's because we were using a specific selection of different donor cell lines within the company and within our processes that we were also able to readily identify and differentiate them from one another by their unique structural variants. We utilize the VIA software in-house to database and catalog both the cell lines themselves, but also the variants we observed across our samples. And we could easily identify several structural variants that were unique and specific to each donor line. And it was in stringently isolating a selection of these variants, we generated our own fingerprints of each cell line we were using. And we selected variants that were consistently observed and conserved for each one. And these fingerprints essentially gave us an additional form of validation for cell line identity, which is another important aspect of cell line characterization. Because we consistently use and test the same cell lines in parallel for product generation, the fingerprints allow us an efficient screen against potential sample swaps that may occur during culture assaying. So for example, for cell line A, you can see we have 5 structural variants on this chromosome that are inherent to the donor of this line. And the presence of 1 or 2 of these abnormalities in another sample might indicate newly acquired mutations, but all 5 together instead strongly indicate that the sample being analyzed is in fact the other cell line, or in some cases a mix of the 2. And this leads into a curiosity we had, which is if we have reliable fingerprints for each line, can we also utilize these to identify both in cultures with multiple cell lines? And to this end, we created some pools of 2 different donor cell lines at different ratios and ran them through different analysis pipelines to assess how reliable the detection would be. What we found with this was very promising, with variants making up even 5% of the DNA in a sample. We were able to consistently detect structural variants of all of our fingerprints. What I've left here on either side is the most extreme example, which is with 5% of the DNA in the sample being of one cell line, but done with the de novo analysis, which is a pipeline that is not optimized for detecting low-level mosaicisms, and yet we were able to always detect some of these heterozygous variants, even down to 0.025 VAF. And whilst we don't routinely generate pools of different cell lines, in the context of our QC pipelines, this has meant there was also potential for OGM to screen against unintentional cross-contamination of one sample with another. In cases where there might be a minor cross-contamination, if one cell line has a higher growth rate, there's the potential and fear that that might grow over time and make up more of the culture. And with this, I hope I've highlighted the extent of verification and screening that we've been able to utilize from OGM. And with all of that said, we have plans to continue exploring, refining, and optimizing the applications of the technology within the company. Because of us essentially consistently using technology for a few years now, we've established a lot of experience and expertise in-house that we have been able to share with external collaborators as well. So if anyone is considering implementing OGM within a similar pipeline or looking to collaborate in regards to utilizing the technology, I encourage you to reach out and to check out bit.bio's website for any information on products. And with that, I'd also like to thank my team at bit.bio, as well as all the colleagues who have contributed to the work. And thank you all for watching.